The origin of cellular life addresses how simple molecules on the early Earth eventually became systems capable of growing, copying themselves, and evolving. Understanding this transition is important because it shows how chemistry turned into biology when natural selection could start acting on living systems. This video will examine three key questions. First, what molecules could replicate and carry out chemical reactions in early life? Second, what environments could have supported these molecules? Third, how did these molecules become enclosed in compartments to form the first cells? Research on RNA and protocells provides a plausible path from simple chemistry to the first evolving systems. One leading idea for the origin of life is the RNA world hypothesis. This hypothesis suggests that RNA came before DNA and proteins and served both as a carrier of genetic information and as a catalyst for chemical reactions. RNA can store information like DNA, but it can also fold into shapes that allow it to speed up reactions, acting like an enzyme. Parts of modern biology, such as the ribosome, still rely on RNA for essential reactions, which suggests RNA played a central role early in life. Higgs and Lehman, in a 2015 paper, explained that RNA molecules may have formed networks in which some molecules helped others to replicate. These networks could increase stability and allow early systems to become more complex. One challenge for RNA-based life is replication accuracy. If copying errors are too frequent, information cannot be reliably passed to new generations. Early RNA systems may have overcome this problem through mechanisms such as grouping molecules together or repeating sequences, allowing natural selection to operate on them. Experiments in the laboratory show that RNA molecules can undergo evolutionary change. Papastavro, Horning, and Joyce, in a 2024 paper, studied an RNA polymerase ribozyme, which is an RNA that can copy other RNA molecules. Over several rounds of replication and selection, the researchers found that the RNA evolved to become faster and more accurate. This demonstrates that RNA alone can support replication, variation, and selection. These experiments are important because they provide a model for how life could have started without DNA or proteins. They also give scientists estimates for realistic rates of replication and error, which help explain how early life could have survived and evolved in prebiotic conditions. The chemistry of life required suitable environments. Pierce and colleagues in a 2017 paper modeled warm little ponds, small pools on the early Earth that experienced repeated cycles of drying and wetting. During dry periods, molecules would become concentrated, making it easier for them to link together into polymers. When water returned, these polymers could mix, separate, and interact. Meteorites may have delivered additional organic molecules to these ponds, providing building blocks for RNA. Such environments may have been more favorable than deep-sea vents because the cycles of concentration and dilution would promote chemical reactions repeatedly, giving RNA molecules many opportunities to form and evolve. These pond systems could have provided both the raw materials and the repeated conditions needed for early molecular evolution. Even with RNA molecules capable of replication, early life required compartments. Schrum, Zhu, and Jostak in a 2010 paper showed that fatty acids and similar molecules can spontaneously form small bubbles or vesicles that enclose RNA. These protocells provide three main advantages. First, they keep molecules together, increasing the chances of chemical reactions. Second, they protect molecules from harmful conditions. Third, they allow selection to act on the combined system rather than individual molecules. Vesicles can grow by taking in additional fatty acids and divide into daughter protocells, allowing RNA inside to be inherited. Fine and Moses in a 2025 paper also describe RNA condensates, droplets of short RNA strands that can serve both as catalysts and primitive compartments. These droplets may have allowed early RNA molecules to replicate and evolve even before membranes formed. Together, these models show how molecules, environments, and compartments combine to create systems capable of evolution, laying the foundation for cellular life. Current research supports a multi-step pathway from the origin of cellular life. First, RNA molecules formed and replicated, establishing heredity. Second, environmental cycles such as wetting and drying helped concentrate molecules and promote polymer formation. Third, encapsulation in vesicles or RNA condensates created a selectable unit where RNA could evolve as part of a system. These steps mark the transition from chemistry to biology and illustrate how the earliest forms of life could have arisen on Earth. Understanding this process also informs our search for life on other planets and helps define the minimal conditions required for life to exist.